Okay, folks, and we're back. So we've got the first, I was going to say the first proper talk. I'd never count my own as a proper talk. So we've got a great one today from a great organisation, CPI. Known them for years, know many of the staff. So we've got Jonathan Rand, who's the team leader at the Food and Feed department or aspect of the Centre for Process Innovation. Jonathan's going to talk today about navigating the alternative protein scale-up journey. Scale-up is the bane of everyone's life. So hopefully we're looking for something interesting here. Jonathan, over to you. Look forward to this. Cheers.
Oh, fantastic, Derek. Thank you for that warm introduction. And thank you for your introduction to alternative proteins prior to this talk. I thought it was a really fantastic introduction. And also, I want to thank the organisers for this event for letting me or giving me the opportunity to speak today. So, to crack on, I am John from CPI. I lead the CPI's Food and Feed team. I'm going to attempt to squeeze what will be still a very brief introduction to scale up, at least from CPI's perspective, into the next 20 minutes. And I'm sure I'll be booted along if needed. So thank you. Right, so we'll start. So briefly, who are we? Well, we are CPI, an independent technology and process development business, part of the high value manufacturing catapult in the UK. And we were set up to help people scale up their processes and technologies and to get them to market as quickly as possible. We don't manufacture anything ourselves. We're only here to help other businesses develop their processes. Now, this does sound cheesy, but our only success is when our customers become a success. So we're always highly invested in our customers' journey. So we've been around since 2004. We're now a team of over 650 people working all over the UK. We're home to over £220 million worth of process development and scale-up assets and infrastructure across a broad range of technology areas, including, of course, food and alternative proteins. My favourite number on this slide is the over £3 billion of private investment we've managed to help our customers and partners raise as a result of them working with us. So back to our topic, navigating the alternative protein scale-up journey from strain development to demonstration scale. In short, we're talking process scale-up. And that pretty much sums up what we do here at CPI. So what do we need to scale up a process? Well, you need a range of technical skills. Process development scientists to make sure everything does what it should. Engineers to turn that science into something more industrially relevant. Also, you need the ability to understand your process from a scalability and economics point of view, and make sure it's going to do what you expect when you get to scale. And also, you need someone to run your plants, and that's really important when you're scaling. Alongside that, you need a whole bunch of non-technical skills as well, and these are often overlooked. You need to have a business model that works, you need to have access to funding, you need the right team in place to deliver a business, not just a process. You need to have the right relationships and networks to fill those gaps that you don't have in your own capabilities. Without access to all of these, you will struggle to successfully scale up. And so where do we start on our scale-up journey? Essentially, we've got some super interesting science and we need to build a bigger version of it. Nice straight line, simple as that? No. Not quite. There are so many questions. Of course, your process needs to work at scale and be efficient and be profitable. But there are so many other questions that we need to ask on the way. For example, how big is the market that you are going into, and how much of that do you realistically think you can take? And you might find that when you get there, no matter how good your product is and how good your process economics look on paper, if the public perception isn't right, nobody's going to buy it. It's that taste and price thing. Who's going to come back for a second bite if it does not taste great or the price point is wrong for your customer? Other questions. Who else is already doing this process? And why am I better? What differentiates me from the competition and how am I using that? And maybe a more pertinent question to this talk, do I have a reliable supply chain that's going to be able to provide the materials I need in the quantities I need when I get to scale and at a cost that works for me? So let's try to break these questions into more simple questions. What should I do? When should I do it? How should I do it? And why am I doing it? My favorite of these questions is why. And this is often the first question we ask our customers when we start talking about the scaling up process. Because there are so many different reasons why you might be doing what you're doing. And depending on what these reasons are and the way you want to go about scaling your process, they could all be different. So for example, if you are building a pilot plant, maybe you will be needing to test some equipment for parts of your process to make sure those elements work. Maybe you're just needing to make some material for testing purposes. Maybe you've developed a novel technology and you need to prove that it works at a relevant scale to unlock the next round of your funding. These are just three possible reasons, but there are so many more, and understanding the motivations behind taking a particular scale up step is super important to make sure you're doing the right thing at the right time. And speaking of the right time, when we do make the next step in scale, or when do we make the next step in scale? Again, keeping it super simple. This is a graph of how scale up risk changes the more time you spend on de-risking it. For me, there are three fundamental process scale up risks. The first one is the process never works. It will never work. Clearly, this is too big a risk and you wouldn't scale it up. But then after that, you've got more nuanced areas where there are decisions to be made based on your risk appetite. Say you have a process that doesn't work right now, but you're pretty sure you can make it work when you get there. If you have a high risk appetite, you might actually go for it. Because if it does work, then you've saved a bunch of time and money in process development. However, if you have a lower risk appetite, then you could spend more time in process development, move yourself down the curve a little, maybe get to a point where it works, a lower risk, and a better chance of a successful outcome. But this is over a longer period of time. Understanding where your sweet spot is on the curve and that of your investors is important to determine when to go to scale. Moving in the right way, at the right time, with the right support network in place is key to success. Now, this is quite a busy slide, but it highlights the different development process capabilities that are often needed to bring a product to market. And in the next few slides, I'm going to give you a very brief overview of host strain selection, upstream process development scale, the downstream process and production of food grade products. We are talking food processes. And so the key thing here is that we need a comprehensive food safety management system and also the right facilities to be able to make food safe products. So where do you start? Well, you start with the choice of strain.
Choices will be restricted.
But especially where your product is not the cell, but a product produced via precision fermentation, we should try to ensure that you're making good early decisions. Is it regulatory amenable? Is there a history of safe use? Can you avoid the GM regulatory pathway? Will it present food safety challenges, such as requiring antibiotics or producing mycotoxins, or maybe allergens? What is the genetic suitability? Is there a genetic toolbox available? How genomically stable is it? Is glycosylation required? And is proteolytic expression going to be a factor detrimental to your product? And there are other technical considerations. The feedstock, tolerance to the final product. Is the product produced in the cell or secreted? Is there a history of scaling of this organism? We next need to consider the upstream development in preparation for the eventual scale-up. Firstly, the selection of feedstocks, where we will consider the carbon and nitrogen sources, the macro and micronutrients, and we'll be working at a small scale. We'd mainly be using micro-well plate bioreactors, such as the biolector or shake flasks, doing straightforward screening and maybe using a statistical design of experiments approach. And there are other things to consider. Cost. Can the feedstocks be supplied as food grade? What about their traceability? And notice we are starting to make key development decisions at a small scale where the cost is limited. However, micro-well plates and shake flasks do not represent a well-controlled fermentation environment. In a bioreactor, you have control over pH, temperature, dissolved oxygen through agitation and airflow rate. Transferring the medium into a batch fermentation process enables you to assess more realistically the performance of the chosen strain on the medium and collect process data with key metrics that include growth rate, product titer, productivity, yield throughout the course of the fermentation. Now, this is a batch culture where you have your growth medium that you inoculate, and other than pH and airflow, it's essentially a closed system that will end by running out of the substrate or through inhibition. Is limited by what you supply the microorganisms at the start. To extend the growth and production to achieve the desired biomass concentration and produce titer, you need to employ a feed. This is a fed batch culture. Growth rate will be controlled by the rate you feed a limiting substrate, and so there is a choice of which limiting substrate. For a biomass fermentation, this will typically be the carbon source, but you'll also need to determine the feeding strategy, whether you're going to feed things in a linear fashion or exponential. At this stage, you optimize your process with consideration of the scaled down parameters. So you do not go beyond what is achievable at scale. Again, notice the process design decisions are being made at lab scale. The next stage is to demonstrate at scale or at a scale, pilot scale, that the process can be fully scaled. At this point, we have de-risked the process and are running at a higher scale to prove out the process, defining it for further scale up and to generate material, e.g. for samples, for testing and formulating into products, and for running representative batches for regulatory submissions. And this will happen on a plant where there is 24/7 operations by a team who are experienced in doing this. Perhaps the key part of the scale-up process to get right is the downstream process. Now, this can contribute up to 80% of the cost of producing a bio-based product. This can require a mixture of DSP methods and unit operations in order to dewater and achieve the desired level of purification, the ingredient specification. Now, the initial stage is from harvesting is that solid liquid separation, the cell separation. And this would commonly use distack or decanter centrifuges, but you could also use things like microfiltration or settling, for instance. Which route that you then take is down to where the product is. If it's in the biomass, then obviously that you will retain the cells. But in precision fermentation, this could be either in the liquid or the solids, depending on if the product is secreted or not. If it is not secreted, then there's a need to disrupt homogenization, high shear mixing, bead milling. Bead milling is very effective for those tough microorganisms such as algae and yeast. In order to achieve the desired product specification and remove undesirables to safe levels, purification and concentration through filtration, precipitation, or chromatography are employed. And then there may be a drying step, and dewatering costs money. Up to 40% of the product can be lost from a precision fermentation through the downstream process. In addition to all the technical side that we have to employ in scaling up, in order to produce food-grade materials safely, it is key that you meet the legal requirements and have sufficient food safety management systems in place, such as a dedicated food safety team and quality manager and a qualified HACCP team. It is industry practice to develop and review a hazard analysis for critical control point plan, a HACCP. And for example, CPI can demonstrate that we have a comprehensive food safety management system in having achieved FSSC 22000 certification. Subjecting your business to audits for FSC 22000 or its equivalents like BRCGS food safety is a great way to build an effective food safety management system and culture within your organizations. And this is needed for scale up and market entry. So I want to leave you with this thought. Recently at a NAPIC event, I heard a comment by Andrea Ariza-Kalora. She's the technical director of Microlab, and she said the following: if you think things can go wrong in the lab, just wait until you start scaling up things. And then there is food grade. Now, I think this highlights why it can be so advantageous to work with organizations like CPI, who not only have the process assets, but a team experienced in developing and scaling processes and producing food grade materials. CPI has the capability, the experience, and the expertise. Not only the assets, but also the right people. Thank you very much.
Thanks for that, Jonathan. That was good. Right on the number. That's perfect.
Oh, wow.
Right, we're getting some interesting questions coming in. So there's one from Chiara, who's at Formo. She's saying, looking at your time versus risk curve, it's kind of like thinking about time versus cost. So how do you balance between going big early or waiting a much longer time for process optimization? I mean, my gut feel is if you're not in the scale up business, you don't know how to scale up. That's the key thing.
Yeah, I think it really matters how well you know your process from the start. If this is a process that is fairly well understood, you're maybe directing your product into maybe a slightly different market, or you're taking different products from the fermentation, then of course that has less risk when you go to scale. It's a known fermentation. But when you're dealing with more complex processes, when you're dealing with things which are much more novel, then that is really the time when you need to be investing the time, I would say, to save you money in the long term. A big mistake is going to cost a lot of money at any of the larger scales. So, yeah.
Yeah. Okay, so one that might follow on from that is actually one from Elvedon at Holac. He's talking about when you're going to scale up, what bits really need physical implementation? I think probably coming to that is the evolution of AI and how much you can maybe do in silico beforehand before you actually jump to steel in the ground and building pilot plants. Are you starting to see an evolution of that?
Well, I'm not really engaged with that too much myself, but there are companies that are at the moment using AI to help develop their processes. I think you can only do so much in silico. You're essentially using models, and models can only tell you so much. And in fact, one of the key things about a model is that it's always going to be wrong. So you always are going to need that certainty of doing an actual wet lab fermentation at whatever scale, just to demonstrate out your model. But also, the people that I would be most concerned with, if you were just taking things via an in silico route and using AI, would be your investors.
Yeah.
They inevitably are going to need to see a demonstration of your process. They inevitably are going to need to see actual samples, physical samples that they can handle, can view, can see, can send for testing, can get assessments on. Sure. So yeah, you are going to have to. There's no avoiding.
You're going to have to jump at some point, basically.
Absolutely.
Yeah. So a good one from Alva Smith at A&A Biotech. You're talking about what do you see as the most difficult aspects for scale up from a technical perspective? So what have you seen? What are the ones that have gone wrong, for example, on big scale up? Again, I suspect what we're talking to is the 4-digit scale ups, really, or the 100 litre and beyond. What are the key? Because if you're getting to that big scale, you're into. I mean, I imagine heat transfer is going to be a big issue just simply in those scales.
Yeah, absolutely. I mean, certainly as you scale, things do change, and you mentioned heat. The removal of heat from processes, because the metabolic burden just increases, and so that becomes a challenge and can be a bottleneck. Also, when you scale, there's also that greater risk of getting contamination. So it's just through the sheer size of the volumes that you're utilizing. So there are definitely issues when you go to scale, and understanding your process and at an earlier stage, I would say, in terms of what risks that you might present as you going forward up the scale, is really important, because then you can, I mean, things like if you do find a system, a process that is subject to contamination, you can understand what type of contaminants there are. It tells you that you'll need to use a particular type of sterile engineering in your process. So there's lots of things you can learn at an earlier scale. So yeah, I mean, things which are obviously changing, the oxygen transfer needed, that might become very critical at scale as well. It's easy to supply lots of air and oxygen at a lower scale, but maybe less so at a higher scale.
Cool. Okay, I think we'll to draw it to a close there. Alvin, you opened Pandora's box of problems there, I think. But I think your man to chat to is Jonathan. His details are there. You can find them backstage. Please do that. Thanks again, Jonathan. That was great. I'm sure we'll talk soon.
You're very welcome. Thank you.
Bye.